I'm sure you've already heard the story that Sinogene Biotechnology, a for-profit pet cloning company based in Beijing, recently announced that one of their beagles had given birth to a live Arctic wolf clone. Yet despite a lot of media attention at the time, much of the bigger picture has conveniently been forgotten. For example, did you know that Maya isn't actually the first wolf to be cloned? It's a story overshadowed by a bioethics scandal that almost completely destroyed one prominent scientist's career, and uncovered a myriad of other problems which haven't quite been resolved to this day. Welcome back to the Dark Wolf Project.
We can learn from wolves, not only about the world around us, but the world within. They occupy a unique place in the land and the mind, in these dark hours at the dawn of the Anthropocene.
Maya's story actually begins with her parent, a supposedly wild Arctic wolf who was imported from Canada to Harbin, Polarland in northern China in 2006. The original Maya reportedly lived for about 15 years, which is a pretty good lifetime even for an Arctic wolf. When she passed away in 2021, the park entered into a partnership with Sinogene to try and recreate her. Exactly why this was undertaken has not been made completely clear. Perhaps the park were unable to source another Arctic wolf, or maybe Sinogene saw an opportunity for proof of concepts. Perhaps a bit of both. To recreate her, Sinogene used a process called SCNT, or Somatic Cell Nuclear Transfer, first pioneered by the Roslin Institute in Scotland in 1996, who famously went on to clone Dolly the Sheep. The process itself, in brief, goes something like this. We first take a somatic cell from the animal to be cloned, and then transfer its nucleus into an enucleated oocyte. That's an immature egg cell minus its nucleus. Of course, the transferred nucleus is diploid as opposed to haploid, and so contains all of the genetic code as opposed to just half of it for the new animal. This means that the egg now contains the information for an exact copy of the parent. Once the embryo begins to develop, it's implanted into a surrogate mother who will carry it to term, in this case a beagle. Sinogene actually created 137 of these embryos, 85 of which were then transferred to 7 different surrogates. Of the 7 beagles implanted, only one actually carried it to term and gave birth to a healthy wolf pup, which was Maya. A second attempt used 99 embryos, 61 of which were implanted into 7 beagles, and one of them developed to term, resulting in the birth this time of a male clone of the same wolf Maya known as Haya. Initially living with her mother, who she very quickly outsized, Maya was delivered to Polarland for display to the public at the end of September. Haya is intended to be introduced when he reaches 100 days old, with the ultimate intention for the two of them to produce offspring. Since neither animal has yet lived with other wolves, the park are quite correctly concerned about introducing them to other animals. Despite the manager of Sinogene referring to Maya and Haya as endangered animals, and Global Times even going so far as to say that Arctic wolves are endangered in the Red List of Threatened Species, Arctic wolves are somewhat ironically the only wolf population whose range is not really threatened by human activity. The IUCN even categorises them with grey wolves as least concern, a contradiction which really raises some questions as to the rest of the story's reliability. Sinogene has also announced plans to partner in the future with Beijing Wildlife Park to build a partnership on gene seed preservation, cloning technology applications in rare and endangered wild animals, which we'll come back to later in the video. During the press conference at Maya's announcement, General Director of Sinogene Mi Zidong, no idea if I'm saying this right, proudly announced Maya's birth as the first of its kind in the world. And although she is the first Arctic wolf, she's not the first of her species to be cloned, or even the second. And this video is about to get a little darker. In March 2007, a group of researchers at the Seoul National University announced that they had used the SCNT process to clone 2 Korean wolves, a subspecies whose taxonomy is still a little bit debated. As with Maya, enucleated dog oocytes were fused with somatic stem cells from the ears of 2 captive wolves, resulting in 4 pregnancies and the successful live births of 2 healthy, completely identical wolves through surrogate mothers. These identical animals were named after the research facility itself, the prefix SNU giving them the names Snowwolf and Snowwolfie, which I guess makes sense. Then, a month after its publication, the paper was pulled from the journal's website. The reason for this? One of the co-authors had just been sentenced due to his involvement in a human cloning experiment. The researcher in question was one Dr. Hwang, and he was considered one of the foremost experts in his field right up until 2004, when he published an article in Science announcing his team had successfully cloned human stem cells using SCNT, something which was thought to be impossible at the time and ultimately helped realise the potential of therapeutic cloning as a treatment. The year following his announcement, however, claims began to surface that he had breached ethical guidelines in oocyte sourcing, including purchasing from the black market. Dr. Hwang immediately resigned, but by now it was too late. His team's results were investigated and deemed to be largely faked. Science unconditionally retracted both of his breakthrough papers, and the deluge of accusations, which included breaching the Declaration of Helsinki, eventually landed the doctor a 2-year suspended jail sentence. Despite this, Hwang managed to retain enough popularity to start up another company, Sinogene Biotech, which still operates to this day. After success with interspecies SCNT on coyotes, his ambitious plans have included cloning the Ethiopian wolf, not to mention red wolves and mammoths. But since then, the company has actually stepped away from the media spotlight. This is where we return to the story of Snowwolf and Snowwolfie, who, like the company's other exotic animals, were donated to local zoos and subsequently forgotten. It wasn't until 2009 that this short article in the Korea Times announcing the death of Snowwolf from an apparent infection exemplified the dire conditions these animals had been facing without enrichment or hiding places. For reference, the WAZA denounces pretty much everything in these photographs as unethical. It gives rise to questions as to just what our moral obligations towards cloned animals are. Are they products or individuals, and how does this affect their rights? Remember Maya, who we started this video with? Controversy surrounds her new home, Harbin, Polarland, which, at the time of writing, is temporarily closed. Photos taken by guests show once again the distressing condition their animals are subject to, with the Polar Bear Hotel drawing the most heavy criticism. It looks like Maya and Haya, through no fault of their own, might just have ensured the continuation of this amusement park. A somewhat ironic twist when, in the words of SOAM's research director, it is the most meaningful way we can use SCNT to benefit society. Reproductive cloning, such as we've discussed here, is still at the exploratory stage. The process itself is still very inefficient. This means it's not only costly, but raises concerns since so many surrogate mothers are involved, and a large majority of pregnancies fail to mature to term. That's not even considering the varied applications, from commodities to conservation. With regards to the latter, introducing cloned animals to a wild population both genetically reduces the genetic diversity of that species, but also potentially risks introducing genetic abnormalities to the population. Hence, Luigi Boitani, an authority on wolves among many other things, argued in a 2016 New Scientist article on the subject of cloning wild canids, "I do not see any canid species in this desperate situation yet," and made it clear that he considers cloning a true last resort. But what about the species it's already too late to save? As well as the irresistible goal of cloning extinct animals from tissue samples, known as de-extinction, There is the very real potential to freeze cells from currently living species. Known as cryoconservation, this method produces a stock of genetic material for later use. Although its application has so far been limited to livestock, there is the potential here to create a gene bank for endangered wild animals. But then we have to consider, would this only greenlight future biodiversity loss by alleviating us of long-term responsibility? The stories we've looked at today are divisive and controversial, yet cloning technology is still in its early days. What are your thoughts on this practice? Do the benefits outweigh the risks, or are we already abusing this technology too much? Perhaps it brings out the best and worst in us. If you would like to join in keeping our large carnivores healthy and wild, why not consider this video's sponsor, the Dark Wolf Project? You'll be helping fund the vital work of 3 worthy causes who specialise in large carnivores, so you get to feel good too. Use code THANKSFORWATCHING for a special discount. If you already follow me on social media, then you've probably heard that I've finally secured a job in real life in conservation and land management. Now, I don't usually do personal stories, but maybe you're trying to start out a similar career yourself, and like me, you don't have a degree, so if you'd like to know how I managed to get into this line of work, let me know down in the comments and maybe I'll come up with something. Okay, that's it for this time, so until the next video, see you soon.